Does anybody have any feedback from last class? Any questions about duplications, rearrangements, translocations? Did anybody find a better way of turning a horse chromosome into a donkey chromosome? We came up with one set of rearrangements that you could use to go from the order of genes on the left to on the right. I know there is one that was better than the one we did last time, because one of you came and talked to me about it afterwards. Hopefully, if you're planning on being a geneticist, you love logic puzzles, because that's basically what genetics is. Here's a puzzle. Figure out how you get from step A to step B. What did we start with last class when we did this? What was the first thing we did? We've got 5 genes on the horse chromosome. We've got 5 on the donkey chromosome, but they're different genes. So the first one was, yeah, we deleted, that was the first thing we did last time. We inferred, we suspected that what happened was that there was 2 breaks to that chromosome and a deletion of genes C and D. So that gets us some of the way to getting the situation in the donkey chromosome where there is no gene C or gene D. What would be another way to do this that's less good, worse, than doing both of those gene losses in a single deletion? What would be worse? What would be less likely? Individual deletions. So you could imagine in blue here that there are 3 chromosome breaks, and that each of those 2 genes are individually deleted. Less likely. So if we have a situation where we have genes A and B and gene E, what can happen now that was better than what we did last class? So we could have, indeed, so we could have, whether or not that's less likely, I'll leave it to you to argue. But we could have a duplication of A and B. So they could duplicate and they could invert at the same time. So this requires no more chromosome breaks. I think the scenario we came up with last time, there were more than 2 chromosome breaks. Here we've just got 2. One there, one there, one deletion, one duplication, and that's it. So I think this is a more parsimonious, which means more likely, more probable scenario than the one we came up with last class. So thanks to those of you that pointed this out. I think this is better than the scenario we had last class, simply because it has fewer steps. What do you think? Any concerns or questions? Yeah?
So they duplicated first and then they inverted? Or?
It wouldn't really matter. So the question was, did the duplication of A and B happen first, or did the inversion happen first? So once again, I guess I would say I challenge you to do things in the opposite order and see if it makes a difference in the end. But when the bottom line is when chromosomes or pieces of DNA are floating freely in the nucleus, we could debate whether or not we even call that an inversion. It is a piece of DNA that turns upside down, but that's not considered a step. So once chromosome breakage happens, those inversions are essentially free. The cell, like we talked about last time, the cell can attach ends of pieces of DNA together in any orientation. Other questions or concerns? Just by way of reminding you of what we were talking about last class. So today we're going to talk a lot about a new technique, polymerase chain reaction. Hopefully everybody watched some or all of the videos I suggested. we do this to detect specific sequences of DNA. So when we left last class, I tried to motivate this with the idea of giving you a couple pieces of corn and you trying to figure out which one's genetically modified and which one's not. And how would you go about detecting any sort of genetic change to any sort of organism anywhere? The most famous version of this, to me anyway, the most fun, maybe we should do this, is going to the grocery store and buying different fish and then determining which species each fillet you bought really is, right? Catch the grocery store in the act, they're passing it off as salmon, but it's really flounder or something like that. So you can tell, I mean, people have done this in classes around the world before, in genetics classes. Everybody go to the grocery store, buy some meat, we'll extract the DNA, we might do a PCR reaction that will help us figure out what species it is that we're looking at. Forensics analysts use this technique all the time, polymerase chain reactions. This is the core technique. CSI, right? People that watch CSI, Law and Order, the people that work in the crime lab over there on campus. It's all based on PCR, and that's why you see geneticists cringe when somebody gets a sample of DNA and then the next minute they're analyzing the results. PCR doesn't happen quite instantaneously. It takes about 4 hours to do this technique. So, you know, I have trouble suspending disbelief when I watch fictional shows. I'm sorry. That's what happens when you're a scientist. So does anybody have any questions about PCR? Just based on what you read in the textbook, really short section on PCR and on gel electrophoresis and on the movies. Yeah.
Why does it get copied after the primers, like where they end? How come it doesn't go beyond that?
Okay. So to answer the question, how does PCR, what is the goal of PCR? What's its purpose? Okay, amplify DNA. Crank it up to 11, kind of amplify?
Multiply.
Yeah, make lots of copies of DNA. To amplify, to make lots and lots and lots and lots and millions and billions of copies of one piece of DNA that you, the scientist, decides. This is just like fish in that sense. Fluorescence in situ hybridization, not salmon. You design the fish probe to help you see what part of the chromosome it is you're trying to find. You design the primers that we use to amplify a particular region of DNA. So let's imagine we've got a double-stranded molecule of DNA, and the point here is to amplify or make lots of copies of one section of this chromosome. So let's say we care particularly about the DNA sequence that's circled in red for some reason. We want to know the sequence of that gene, so we're going to make lots and lots of copies of this piece of DNA to sequence it, to identify the sequence of nucleotides. So in addition, to do PCR, at its most simple, we're going to work toward the answer to the question eventually. What do we need to have besides crime victims' DNA, say? So this is called template DNA. You could think of it as a forensic sample or DNA that was extracted from fish at the supermarket, something like that. We need one other type of nucleic acid besides the DNA sample that we're trying to make copies of, and those are the primers. Those are single-stranded pieces of DNA that are complementary to the part of the template we're trying to copy. In other words, if we zoom in, one primer might be complementary to those two nucleotides. So let's say that's an A and a C in the DNA. The primer sequence would be something that would base pair with that template. So if the DNA is A and C, what are the nucleotides on the primer?
TG.
Yep, TG. So sorry, I should have done the A and the C in black to keep up my color coding. So A and C are DNA sequence. So we'd make a complementary sequence of this primer, T and G. Let's see. We can add in some polarity too. So the reason I wrote this little half arrowhead on the end of the primer is because that's going to be the direction that DNA synthesis proceeds, from left to right. And as we learn later in class, when we talk about DNA replication, you might already know this, DNA replication proceeds from the 5' to the 3' direction. So when polymerase chain reaction occurs, what happens is an enzyme, that's the third component. We've got template DNA, we've got primers, that's the second part of a PCR experiment. What's the enzyme we add to synthesize the DNA? Yeah, DNA polymerase. So that's an enzyme. It's a protein that does the job normally in our cells as well as we do it in this case. We add this enzyme, DNA polymerase. So we'll call that DNAP polymerase. We'll grab onto the 3' end of the primer and move that direction, and it adds in the extra nucleotides to make a new copy of DNA. So it's reading, I'm going to make up some more nucleotide sequence now, again of the top strand of DNA. So in this tube that we've added the template DNA, the primers, the enzyme to, DNA polymerase grabs onto the 3' end of the primer, and then to the end of this primer, it's going to add, what's the first nucleotide polymerase incorporates? It's whatever is complementary to that strand of DNA we're copying. And then it's going to add a, and an A, and a T, and a G. And that makes a continuous piece of DNA that starts with the primer and continues. So polymerase keeps adding nucleotides onto the end, the 3' end of this growing piece of DNA, moving from left to right. It's like that insurance commercial. DNA polymerase, it's what it does. Okay, now, how many primers are there in a PCR reaction? How many different primers are there? 1, 2, 3, 4, 10, 20? 2. So where do I draw the other primer? It's going to be on the other strand. It's going to be down here somewhere. And it's going to be synthesizing that direction. So there's its 5' end, its 3' end. So let's say the sequence of that primer from 5' to 3' is T and then A. What does that mean that the sequence of DNA that primer will be base paired to, the bottom strand here, is going to be? The A pairs with T, T pairs with A. So we've got another DNA polymerase enzyme, because as always we throw in, as in most experiments, like with the fish probes, we throw in lots of extra enzyme, lots of extra primers. So there's tons of molecules of these, not literally tons, in the test tube, the DNA, the primers, the DNA polymerase enzyme. So at the same time, one DNA polymerase enzyme is synthesizing the top strand using the primer on the left. A second DNA polymerase enzyme is here bound to the 3' end of the second primer. In which direction is it going to move? Left to right or right to left? Well, it's going to start here, so which direction is it going to have to move to add new nucleotides? That direction. Okay. So you can see up here in the upper left the sequence of the top strand of DNA, right? From left to right, TAC. So what's on the bottom strand underneath it? What's the sequence of the bottom strand of DNA underneath those 3 nucleotides? The complement. This is just chromosome we're talking about. So they're base paired together. So what nucleotides are going to get added in under here as DNA polymerase synthesizes from right to left on the bottom strand? What goes in blue here? C, A, T. So the synthesis of the top strand keeps moving down the top strand of the DNA from left to right, just goes and goes and goes and goes. As long as there's chromosome to copy, the enzyme keeps copying the black strand and making the blue strand, its complement. And the same thing is true down here. So from each primer, the DNA strand only gets copied in one direction. Just goes and goes and goes and goes and goes. So getting back to the original question, why is it that we only make a copy of a specific part of DNA? Why does this process only limit us to making a copy of a discrete piece of DNA? How do we define which part of the chromosome we're making copies of? It's in your control.
Where we put the primer?
Exactly. It's where you've designed the primer. So, in other words, if I zoom out a little bit more, what happens there? Does a new strand of DNA get synthesized left of the 5' end of the primer? What about down here? So there's more DNA, there's more chromosome down here. What happens in there? Nothing, because those are the parts of the chromosome that are upstream or 5' of the primers. So they never get copied. And when you do round after round of PCR, how many cycles of PCR do we normally do? It's like 25 or 35. You wind up making how many copies of this DNA? Billions, trillions, something like that. Billions, literally, of copies of the single molecule of DNA you start with. 4 hours later, you've got literally billions of molecules of this DNA strand. And they're limited by where you put the 5' ends of the 2 primers. You only make copies of that part of DNA. Sure, there's a little bit of copying that goes on out here, because I told you that strand, that blue 3' end of the synthesized strand will keep going. But that's only one copy of DNA. You have to watch those videos if you really want to get the sense for why PCR limits the size of the product that you produce. In subsequent steps, all of the PCR products are limited to the flanks where you put the primers, the 5' ends of the primers. So ultimately, after you're done with 4 hours of PCR, which is great because it used to be you had to do this by hand. You'd have different water baths set at different temperatures, and you'd have this huge flask of liquid, and you'd hand hold it in one for 30 seconds, then you'd move it to another temperature for a minute, and then you'd move it to another temperature for 3 minutes, and then you'd move it back, and you did this all by hand for hours and hours and hours. Now we have machines that have one water bath, essentially. It changes the temperature for us automatically. So you do it in 4 hours instead of hand holding these tubes and spending days just changing the temperature of these reactions to make this process work. After this is all done, you've got trillions of copies of this DNA. Most of them are the same size. They're this size. It's exactly the distance from the 5' end of one primer to the 5' end of the other primer. So the length of the amplified DNA, that's how it's defined. Why do we care? So you made a bunch of copies of DNA that has one sequence. As I hinted at before, it's useful because now we've got lots of copies of this DNA. We can actually do things like sequence it. It's really difficult to sequence a single molecule of DNA, but if you've got millions or billions or trillions, it's easy. There's something else we can do even more simply than sequencing. And that has to do with agarose gel electrophoresis. You remember why we stain chromosomes when we have them on a microscope slide? What's the purpose of staining them?
Find out the location.
So we've got fish, helps us learn the locations of molecules. Those have fluorescent molecules on them because otherwise we can't see where the fish probes are. Why would we band or put that chemical DAPI on chromosomes?
Mass homologous.
Pardon?
Mass homologous.
So to actually see the chromosomes, for example, banding then lets us match homologous chromosomes together. But we can't see DNA unless we stain it. Sometimes all we want to know is what's the distance between the two primers, which is crazy. But we determined the length of the amplified DNA product experimentally. And that's agarose gel electrophoresis. And I meant to have assigned, but I didn't, so I've got the link at the end of the slide deck here today. A short video again on how you do agarose gel electrophoresis, a new technique, to analyze how big the PCR product is you made. But you read about this in the textbook. So you PCR amplify a bunch of molecules of DNA. You've got trillions of them. How do you tell how big they are? How would you know? How would you measure experimentally how long those pieces of DNA are?
The location, and it's on the gel.
Okay, so you have to do gel electrophoresis. So very briefly, what's the principle of gel electrophoresis? Why does it work? It lets us figure out how big a piece of DNA is. What property of DNA allows us to electrocute it and figure out how long it is? What's the charge on DNA? It's negatively charged. All the phosphates along the backbone of DNA have a negative charge. So DNA has a negative charge. Poor DNA. What a pessimist. And it's Friday too, it should be happy. So what happens if we apply an electric charge across a sample of DNA? Which direction will it move? Will it go to the negative pole or to the positive pole if we have a voltage? Right? So opposites attract, the DNA is going to move in this direction. Okay, so far, I've hopefully convinced you that you can make DNA move by shocking it. Doesn't really help us figure out how big a piece of DNA is unless you load it into an agarose gel, which is basically a piece of jello that has some holes, which we call wells in it, little depressions, where you can add liquid samples of DNA. So you can add some PCR product, those copies of DNA, into one end of this piece of jello. Then you apply an electric charge. The DNA in that well, which I'm going to color red here to follow it, is going to move from left to right. Negatively charged DNA is attracted to the positive terminal. So what's this going to look like after half an hour? Where am I going to find red in the gel? Pardon? Towards somewhere, yeah, somewhere between left and right. Somewhere towards the positive terminal. So let's say 30 minutes later, that's where all the DNA is. It's moving slowly from left to right towards the positive charge. How big is that molecule of DNA, or those molecules rather? These are all the copies, one chunk of DNA that's been multiplied trillions of times. There are trillions of copies of DNA there that you can see. They're at one spot on the gel. How big are they?
You compare it to a KB lab.
Sir?
You kind of need another reference piece.
Right, we need to know, yeah, so we have a relative idea of how big they are, but we need some sort of standard. We need some molecules also on this gel of DNA of known sizes. a positive control, say. So in this other well, we load a mixture of synthesized DNA molecules we affectionately call DNA ladder. You could also call it a molecular weight standard. There's a cocktail of pieces of DNA that we already know what sizes are in that solution. So let's say we've got molecules that are 100 base pairs, 500 base pairs, and 1 kilo base pair, 1,000 base pairs, all mixed together, and we add them in this other well in the gel. And we subject them to the same electric field, the same electrophoresis. And you see this. So the black now are the bands of DNA that come out of the ladder well. So those are the 3 different molecular weight sizes that are in that standard. How big was our experimental piece of DNA? How do we tell? Okay, so this is one other principle you have to know about PCR. We've talked about the components of a PCR reaction, all the things you have to add together, template, primers, polymerase, and some other things as well, which aren't as important. We've talked about DNA is negatively charged, so it moves towards the positive terminal. And the last thing you have to know is which piece of DNA moves farther, faster. Is it the small one or the big one? Okay. So with that in mind, what's the molecular weight of that band? Yeah, you've got 3 choices, 100, 500, or 1,000. You just said the smaller one moves farther, so that's the 100 base pair. That means this one's 500, and that one represents molecules of DNA that are 1,000 base pairs long. So how big is our experimental piece of DNA? It's about 500. Great. So I'd like you to think about how you might apply that technique and what results you might get if you had, let's say corn A is the transgenic corn. It's got a chromosome, and in that chromosome somebody has inserted a insecticide resistance gene. And here's the unmodified corn. It doesn't have that gene. So think about, maybe for next class, how would you use PCR to detect the presence or absence of that gene? If you go to the grocery store and you take samples of produce and you want to know are they transgenic or not, what would you need to do? And what would the Agarose gel look like? It's a thought experiment for next time. Now in the last 20 minutes of class, we've got a couple of activities. So we're going to do a few things at the same time. First, I'm going to tell you about what I'd like you to think about for next class. There's a brief movie to watch. I'm going to post the study guide for exam 1. It's written. Thank you for all of you that contributed questions. It was really good. So the study guide PDF I'm going to post on Google Classroom immediately. Here's a link at the bottom to another video on gel electrophoresis in case you want more information about how gel electrophoresis works. So the last 20 minutes of class, we're going to do 2 things. One thing is take a mock exam. So think about it as study guide version 2. It's not for points, but it is to make sure that you know how to do this process in class like we were taking a real exam. That's why it's called a mock exam. At the same time, our guide on the go can circulate through class. If anybody's connected still to the Fresno State network, that one's going away at some point. We want to make sure everybody is connected to the Eduroam wireless network. So those of you that are still on Fresno State or Bulldogs, good point, we want to try to move as many of you onto the new Wi-Fi network, Eduroam. So first thing to do take the practice exam. When you're done with that, if there's still time in class, then talk to Emily about getting onto Eduroam if you need to. So here's how this is going to work. I'm going to post this exam. So pretend this is next Friday, a week from today. You come into class, this is what's going to happen. I'm going to walk over here. I'm going to assign the mock exam. I press assign, it gets sent out to you. So just like every other PDF annotation thing we've done in class so far, in your Google Classroom, I'm going to start doing a lot of those. All of you get these notifications that I just posted a PDF. This is your exam. You have 20 minutes, in this case, 50 minutes a week from today, to annotate the PDF and attach the annotated PDF back to Google Classroom. Not screenshots, not emailing me anything. Right, what we've been practicing this whole time. Annotate the PDF, attach the annotated PDF back to Google Classroom. So work on this now. It's not due, really, it's not due at 11:50, but the goal is do these 2 questions to practice, see if you can get this into me by 11:50. By Monday, I will give you feedback on whether or not you were successful, and that gives you all of next week to work out any more kinks before we hit the exam, when this will be real. Okay? So work on this. When you're done, we can get you onto Eduroam network, and we can also answer tech questions if you have issues doing this process. Okay? See you next week. Stay here, do this activity, and I'll answer questions if necessary.
I don't see anything attached. Oh, never mind.
Yeah, sometimes that's why I like doing this now, because the same thing happens next week if I don't do a practice exam.